talk today, focusing on the sort of basics of YAN, and then I'm going to go over some research we've been doing with primarily Riesling and tell you why that's relevant in other parts of the world too. And then I'm going to present some data on YAN and some of the cold hardy cultivars. So we'll do 20 minutes of the basics and a review, and then we'll take a break for questions, and then I will go into some of the data that we have and some of the practical applications of these things. So let's see if I can advance my slide. Maybe not. Kristen. Okay, awesome. All right, here we go. You all are aware, if you are winemakers, and I'm sure most of you are, that we need to feed our yeast a balanced diet. And we tend to think a lot about these two nutrients that the yeast needs, the fructose and the glucose, those sugars that it converts into the alcohol that we desire. But we don't as often think about the other things that we need, namely YAN, the yeast assimilable nitrogen that the yeast also needs in order to metabolize those sugars and kick out the ethanol that we're looking for. And when we talk about YAN, it is actually an additive. It's an additive formula. YAN is the total of your ammonium ions in your must and your PAN, which is primary amino nitrogen. So there are other nitrogenous compounds in the grapes, but what we're interested in are these two, and combined we call them YAN. So those have to be measured separately. Go into a little bit more detail about these two components. This is your chemistry lesson for the day. This is the structure of the ammonia ion. And what's important is that that nitrogen there in the middle that the yeast need is only connected to hydrogens, which makes it very easy for the yeast to assimilate. Those hydrogens are kind of negligible. So what we are looking at here is ammonia, basically. We call it ammonia ions because it doesn't scare people as much when we talk about having ammonia in our juice. The more complex nutrient is the PAN, the primary amino nitrogen. This is organic nitrogen in contrast to the inorganic nitrogen from the ammonia. And it's also called FAN, which is free amino nitrogen, or assimilable amino nitrogen, or alpha amino nitrogen, depending on who you're talking to. So for our purposes, we're going to talk about PAN because it sort of makes the most sense. And if you're curious why PAN, this is an example of a primary amino acid. And you can see, as with the ammonia, that nitrogen is just hanging off the bottom there. So it's easily accessible for the yeast when it comes in to chomp that off. The other types of amino acids that are in grapes are a little more complex. You can see in this structure that nitrogen is embedded in that ring structure. It's not free like the primary amino nitrogen. This is a secondary amino nitrogen, and it therefore cannot be chomped off like the primary amino nitrogen, so it's not accessible to the yeast. So cultivars that have a lot of proline, for instance, like Cabernet Sauvignon, have some amino acid that's bound up that yeast and other microorganisms can't get to. So what do we have in our must? Well, it depends on the cultivar. We do see a lot of cultivar variation. Viticultural practices, although that's not a part of our talk today, but changing your YAN by viticulture alone can be a little tricky, although there are folks who are working to streamline that. Season and a million other things. We have really yet to find sort of one or two or even a handful of predictors of variables that we can measure and predict YAN content from them, which is a bit of a downer because it would be a whole lot easier if we could. So there's nothing in the vineyard that gives us a predictive parameter. You can't take a petiole test and get an idea of what your must YAN is going to be because there's so many things going on in the grapes that before it actually gets into the must that you don't have any good correlation. For fermentation, we usually say that the yeast are going to need between 150 and 200 milligrams per liter. You can get in all kinds of arguments with enologists all over the world about this. Some folks say, oh, well, really 140 is all you need. There are folks who would push to have as high as 500 milligrams per liter. I think most of those people are sort of backing down a bit. But most of the winemakers that I talk to or enologists I talk to are comfortable in this 150 to 200 milligram per liter range. So if you have a YAN deficiency, and most people do agree that you're deficient if you're less than 140 milligrams per liter, you get stuck or sluggish fermentation. Your protein synthesis slows down quite a bit. This damages your cell membrane. It damages the cell's ability to reproduce, so this is not something you want to happen in the beginning of your fermentation. And also, it damages the cell's ability to build the protective factors that it needs later in fermentation. So if you starve your yeast early in fermentation, you're not allowing them to develop the protection they need in the end when the alcohol is quite high. It also impacts your sugar transport enzymes, which slows down your fermentation. This is a limiting step, so if they're slowing down, you're not going to get good fermentation. And you may get amino acid degradation. The yeast cell essentially starts to cannibalize itself if it doesn't have the ammonia or nitrogen it needs coming in. It has to start breaking down and reassembling its own portions of its own cell, and that's not good. That's when you get H2S production, and that's when you get this lovely skunky smell, which is what we normally notice first, other than just the slowness of the fermentation. The flip side of that, there's some other causes of stuck fermentation. However, don't always assume that it's just your yeast. Your temperature being too high or low is the first thing you want to check, of course. Inadequate oxygen is something that folks often don't think of. Sometimes just a quick aeration early in fermentation will help. Later in fermentation, not usually, but early in fermentation it can help. And sometimes you just have a combination of factors, with a little bit of nitrogen deficiency and slightly low temperature. These things can add up pretty quickly. So don't always assume that you just need to throw nutrient in your yeast. Think about it and check some other causes as well. All right, so the flip side of yan deficiency, more yan deficiency. Yan deficiencies also impact your sensory profile. There are about 23 compounds that are impacted by nitrogen availability. The correlations are not always direct, but some are. Monoterpenes, which we find in the crescent. Also, if you are in New York, things like Gewürztraminer, Riesling, and muscat, some of the aromatic whites, can be impacted if there is not enough yan to build those compounds. There's an inverse relationship to higher alcohols. So if you have low yan, you tend to have higher alcohols, more higher alcohols, and these, small amounts can be floral, but you get too much and it starts to be waxy and just unpleasant. Also acetic acid, so you can get some levels of VA just because you don't have enough nitrogen, because those yeast are sort of struggling. Other compounds like esters, and esters give us all of the nice floral and fruity notes that we like, especially in young wines. These sort of vary by compound, so we can't say, well, you're going to get a decrease automatically. You may get a decrease in some and an increase in others, which may lead to, especially in aromatic whites, it may lead to aromas that are still there. It'll still be quite aromatic, but it won't be like varietally appropriate. So that's work that is still being done. There's a lot we don't know about how these metabolic processes work. All right. Now, finally, the flip side of deficient yan is, of course, excessive yan. That's greater than 400 parts per million. And generally, unless you're one of those folks who feels that it should be more than that, but usually 400 is what we're talking about. The biggest danger, of course, is that you can have rapid fermentation. That can get your heat too high. Your yeast can be problematic, could be unhappy because of that. It also will give you a simpler aroma sometimes. We don't have good scientific data to prove this, but plenty of empirical data, so that's some work that we're interested in looking at. You also have potential for excessive biomass buildup early on, and that can cause problems later. You can end up with a stuck fermentation later because you don't have enough nutrients for all of that biomass. Ironically, in addition to getting acetic acid formation with low yan, you can get it with high yan. And you can get some microbial instability, and this is one that we worry about quite a bit, because if you're leaving all this stuff, the yeast eats everything it needs and there's stuff left in the fridge, then the microbes can come in and clean that out. But if you clean out that fridge before the yeast get out of there, then you'll starve your microbes to a little bit. In addition, we're becoming more concerned about the potential for ethyl carbamate production. This is a carcinogen that can occur in wines that have high yan, especially if there's residual yan. It is regulated in some countries. It is not yet in the U.S., but the way these things tend to go, we start with. Thank you. We start with a few countries, and sooner or later everyone is regulating it, so that's something that's on the horizon we need to think about. All right, so if you think you have low YAN, or if you just aren't measuring your YAN and like to add nutrients just in case. And this is sort of the downside of a webinar. If you were sitting in a big room right now, I'd have you all raise your hand and tell me, A, if you measure your YAN, and B, if you make additions without measuring your YAN. It's always interesting to see what kind of populations we have in each group. You first need to know which your initial concentration is. Before I said 140 and below is deficient. If you want to play it really close, 150 is like the minimum you need for fermentation. Most vendors and most winemakers will recommend 200. Although if you look at the chart down here in the lower right-hand corner, you'll see that that varies by Brix. So if you're working with some of those Riparia-based hybrids where you get very high Brix, you're going to need to up your nutrition requirements to meet that. And this chart is one we pulled directly from the Scott Labs catalog, and Lallemande has got some good information too. There are other vendors who do, but I've just been pretty impressed with the way they present that. So Brix is important, temperature is important. It's not as important as Brix. The amount of oxygen will impact what you've got, and yeast selection is very important. This, for instance, is a chart that Lallemande has put together. It just gives you an idea. They show a range of their yeast and the nitrogen needs that these yeasts have. And you can see that down there on the far left, that S1 needs very little nitrogen in order to efficiently consume sugar, and the ones on the right need a whole lot of nitrogen. And they roughly break those down into like 6 categories. Most yeast vendors will tell you whether their product is a low, medium, or high nutrient yeast, and it's important to pay attention to that. This is another example. You can see this is an Enardis product, and you can see sort of the ways to look and see what you need for this particular yeast. Is the part I've highlighted there. It says it needs a high nutrient addition to require good fermentation rate and full expression of yeast aroma. This one, they say, under enological effects, has low potential to form H2S, but if you really starve it, you might get some more anyway. And you'll see it needs adequate nitrogen to produce high levels of fermentation esters. So the yeast manufacturers sort of know what they've got, and they will tell you, but you need to pay attention to that and think about your ability to measure YAN and your ability to supplement in order to best suit your yeast. So the types of additions we can make are first diammonium phosphate, and if you remember, we talked about 2 types of YAN, the ammonia and the primary amino acid. Well, the diammonium phosphate basically gives you ammonia in combination with phosphate. So it is your inorganic. Because it's inorganic, it is rapidly absorbed. We kind of like to say that this is sort of the junk food of YAN because it's very quickly absorbed. It has no other micronutrients, so you're not getting, in a great must, in addition to the nitrogen, you're going to have all kinds of small amounts of vitamins and minerals in there. This is just straight ammonia. The legal limit in the U.S. is 8 pounds per 1,000 gallons, or 960 milligrams per liter, so that's something you need to respect. There are some issues with all DAP additions, though. It's just like having too much YAN. You can get rapid biomass buildup because the yeast will eat this first. They'll preferentially suck up the DAP and will sometimes leave some of the ammonia or the amino acids that are in the must until later, and because it's built up such biomass, it sometimes has been hampered in using that complex nutrient later. So too much DAP in the beginning could hamper uptake later. You also can get increased glycerol production. And while some of you might think, oh, that's great, that gives me more mouthfeel, it really isn't. It's a detrimental problem. It means your yeast is a little bit stressed. And as with a rapid fermentation, you're going to get a simpler aromatic profile and less ester development. Now, this again is an area where there's more research going on. We're not exactly sure how the different types of nitrogen impact flavor formation, but that's an area where people are starting to do a lot of work. I'll talk about it briefly at the end of this presentation. So the other types of additions are called complex nutrients, and they usually include DAP mixed with some assimilable amino acids, some FAN, and other vitamins and minerals. So these are going to include organic amino acids. They are more expensive because they're these formulated blends. They're all commercial secrets. Usually they don't tell you exactly what's in there. They will give you recommendations for use, and they're usually used more sparingly because they are expensive and because some people are kind of uncomfortable because they're not entirely sure what all is in there. Usually your vendor is not going to tell you everything because they have to make money. They've got their company secrets to keep. But they should give you an idea of when they should, what yeast they're good for and how they should be used. Covered that. So, whoa. How do we time additions? Well, there are 3 times when we usually can add additions. The rehydration nutrient is the first one. This is when you mix in when you are rehydrating your yeast. It is usually taken up rather quickly. Your yeast isn't stressed. There's nothing to bind with it, and you're not competing with other microorganisms. So I'll say this now, and I'll repeat it again. For rehydration nutrients, you have to use a nutrient specifically designed for rehydration. If you use DAP or some of the other complex nutrients, you're going to poison your yeast, basically. I'll talk about that more. The second time for addition is at the end of lag phase. That's usually 6 or 12 hours post-inoculation. Some people will say, well, it's at about 16 degrees Brix, but that kind of depends on what their starting Brix is, which can be quite high in the Vitis riparia. At this point, the yeast have gone through a rapid period of reproduction. The stress is starting to get to them. They need a little bit more food. They need more vitamins. And they've depleted some of their key micronutrients. And the third time is at about 1/3 sugar depletion, so maybe closer to 10 or 8 degrees Brix, depending on what you started with, again. And this will aid the development of those survival factors, the cell wall components and other things that the yeast need to survive at the end of fermentation. So if you're just going to add one addition, it's usually recommended that you add it 1/3 of the way through fermentation. And again, you're checking your Brix to decide when that is. 2 additions, one right at the beginning of fermentation, right at inoculation, 6 to 12 hours after inoculation, and one 1/3 of the way through. And if you're going to do 3, you're going to do same as 2 additions plus the rehydration nutrient. So this, again, this is Scott Labs, not that I get no money from Scott Labs, but they do some nice graphics, and you can find this on their webpage to give you an idea of what you would want to do with low, medium, and high YAM there on the left, and then the types of additions you would like to do. And they also, at the bottom here, you can see a nice schematic of that lag phase, growth phase, and so on with yeast biomass. So I said I would mention this, and I'm going to. They say you only remember 20% of what you've learned, so this is part of the 20% you should remember. You do need nutrients at rehydration. They're often recommended to get that, it's like a head start for yeast, get them off to a good start because they'll protect themselves later in fermentation, but only use rehydration nutrients because these yeast, when they're dry, they're very delicate. Their yeast membranes are not very strong, so they're just sucking everything in that you give them, and if you give them too much DAP, they can actually, too much nitrogen, they can actually mess up their internal cell mechanisms, and you're in trouble later. So it's kind of like the little kid who thinks that, beach sand is a tasty treat. We've got to limit their nutrients early on. Some other things to consider. If you are making a light wine, therefore no skin contact time, doing a 24-hour juice settle so that you get the gross lees out really helps pull out some of the things that can inhibit your yeast, like pesticide residues and heavy metals. These things can cause stuck and sluggish fermentation that aren't related to fermentation or nitrogen issues. If you excessively clarify your juice, if you centrifuge, for instance, I'm sure a lot of people don't really do that, but if you do, or filter, or add too much bentonite, you can remove some of the micronutrients, some of the vitamins and minerals that were in the juice, and it's hard to get those back even if you use a complex nutrient because you're not sure what you've taken out. And finally, for spontaneous fermentations, if you are doing some indigenous or spontaneous fermentations, you likely are going to need more YAM because you've got more things in there competing, and a lot of the wild yeast have higher nitrogen needs, and that's why having low residual nitrogen after fermentation prevents spoilage problems. But that's something you need to think about. You might want to up your information. Up your addition. So at this point, I think we're going to pause for questions. I'm not sure how we're going to do this. Okay, this is going to be interesting. All right. So the first question is, can oxygen levels in the fermentation be monitored with a dissolved oxygen meter, and is there a recommended level to maintain? You know what, Tim, I do not know the answer to that, but that is one that I will turn over to Gavin and Chris, because that is their area of research, and we will make sure that we get that information. Patrick asks, how close are we to having a rugged, low-cost, continuous probe to measure YAN, or am I just dreaming? The answer is yes, you are just dreaming at this point. The best way to measure YAN, I'll talk about it in the second half, but there is really no probe. There's a nice probe for ammonia, but that does not measure PAN, just the ammonia, and you usually need to know both of those. In fact, PAN is often the determining factor. So we'll talk about the ways we can measure it, and I'm sorry to burst your bubble there. David Pop asked, are skin fermentations naturally higher in YAN due to the presence of all skins, etc.? That's a good question, David. Nitrogen compounds are concentrated in the skins, and so in a red wine fermentation, you're going to get some extraction of YAN, but a couple of studies have been done on this. The one that seemed to be the most robust was on Pinot Noir, and they actually found that even though there was a little bit of a small increase in YAN and the skin fermented Pinot Noir, in measuring it just after crushing was okay. So with whites, we usually measure YAN once we've crushed, pressed, and sometimes even settled the juice. So when you have a nice clear juice, you measure your YAN. With the reds, you would crush. Make sure you've got that must mixed up really well, and then pull a sample from the must. So you don't need to do, I mean, if you're going to do a skin contact time prior to fermentation, you might want to go through that that cold soak and then check it before you inoculate. But other than that, there's not a whole lot you can do. You're not going to get so much leaching in during fermentation that it's going to affect any additions you make. Rob asked if in lower bricks, such as an apple juice, does it require less YAN? That is a good question. I only work with grapes. I think I wouldn't feel safe going below 150, just because we start to then just be missing some things that the yeast need. If you have never added nitrogen to your apple juice that you're fermenting and you don't have a problem, I wouldn't start adding it now. Any other questions? Okay. Does the process of hot pressing red wine grapes add more YAN to the must? That's a good question. I don't know that study has been done, and I don't know if you get denaturation. I don't know. I don't know the answer to that. We'll see if we can find that. Matt says, almost all of my H2S fermentation problems come from white varieties I get from the Finger Lakes, such as Seyval, Cayuga, and Traminette. Will high drop conditions affect YAN in grape juice? Ignoring your second part of that question for a minute, I am not at all surprised that you're potentially getting low YAN and things from Finger Lakes. We have looked at lots and lots of grapes here, and we almost always have low YAN. So if you are getting, anybody who's working in the Finger Lakes or who's getting juice from the Finger Lakes is probably going to need to make additions. Drought stress can impact YAN, but we don't know exactly how at this point. Dennis asked, comment on bentonite addition to juice. The short comment to that is, why would you do it? You are really risking pulling out a lot more good stuff than bad stuff. So unless you really have a problem, like you've got a lot of rot or something like that, bentonizing juice is usually not recommended. If you have specific questions about that, drop me an email and I can give you some information. Tim says, can you measure YAN in advance of harvest to help plan out the fermentation additions before crushed? Oh, Tim, such a nice segue. We're going to talk about that after the break. I know exactly where you're going, but thank you. Dorothy O'Brien. Hi, Dorothy. I haven't seen you in a little while. Do I hear you saying that you should use a rehydration nutrient regardless of the YAN measurement? Thank you for that question. That's a good clarification. No, if your YAN is high and you do not need to make an addition, I would not use a rehydration nutrient. However, I would talk to your vendor about that. Granted, they probably are going to err on the side of trying to sell you more products, but depending on the yeast strain, especially if you're using some of the specialty yeasts that have some finicky requirements, you may need a little bit just to get them going. But Dorothy, if I remember, you're in Iowa, and Iowa traditionally has very high YAN, and so you probably don't need to worry about it. Frank says, are standard tests of yan accurate post-fermentation so we can determine if we're adding too much? Yes, you can certainly do, well, standard tests. I wouldn't do formal titration ever, but we do seem to get good results from spectrophotometric analysis as long as you were doing that analysis correctly. And Randy asked, what about yan for late harvest and ice wines? Those are going to be really stressful conditions. I believe that Debbie Inglis might have developed some specific recommendations for those late harvest wines. I don't know off the top of my head, except that you're going to want to look at the sugar content and your yan and probably bump that up to match the bricks that you see. So, what do you think? All right. Okay, I think we blew through a lot of questions there. I hope that wasn't too fast. I'm going to take a little sip of tea so that I can keep talking here. And we'll go on to the second half of this talk. So my colleague Amanda Stewart, who is the new enologist down at Virginia Tech, for her dissertation at Purdue, did some really great work where she looked at yan by species and by state. And so this, as far as I know, let's see, if you haven't muted your microphone, I'm hearing some feedback, so if you guys will just check, please. This is the only study that I know that is a comprehensive look at the states and the cultivars that we're dealing with in most of our cold climate areas. So it's just a starting point, but she has agreed to let me share some of this, so I'm excited to go through it. So one of the things she did was just make a massive survey of all of the types of grapes she could get a hold of across the Midwest and the East. And so you can see in this chart on the left where we've got a sample of 1,701 different juice samples. That's what grad students are for. I'm pretty impressed that she did all those measurements. She has some really nice, robust statistics and can tell us that there are significant differences by species in yan. And so if you look at this chart, see the Vitis vinifera, the traditional wine grape cultivars, average yan per milligram, and this is across the Midwest and the East and across all cultivars. So we're making some huge generalizations here. But in general, Vitis vinifera is around 180 milligrams per liter. You can see that Vitis rotundifolia is lower than that. Labrusca is generally higher. And so those of you who are working primarily with Elmer-Swenson hybrids, this Vitis labrusca number is probably going to be most relevant to you. So in general, these species tend to have slightly higher yan. That's going to vary by place, of course. The Vitis estivalis, which is a background grape that's often, or excuse me, an ancestor grape often used for interspecific hybrids in the Midwest and here at Cornell, is a little bit lower, 170. And then she grouped a bunch of them that couldn't be identified by species into interspecific hybrids, and you can see that is generally higher. Now this is a huge, broad sweeping generalization, but it is interesting that with the type of numbers she had, that she was able to find statistically significant difference in these interspecific hybrids, and that they're generally higher than vinifera. So when you're thinking about additions, you might need, and you just do prophylactic and don't measure, you might want to think of backing off on these hybrids. More importantly, you might think about measuring your yan if you can. I thought it was also interesting, she surveyed by state, and again, this is going to be all of the cultivars that are grown in the state. But I think we can still draw some conclusions here. If you look at Minnesota, Iowa, and South Dakota, I feel pretty safe in assuming that those three states are primarily growing hybrids, and they're often growing the Riparia-based hybrids from the University of Minnesota. And you can see that you see a significant difference from state. And these numbers are rounded off because they're, there's quite a bit of variance there, but they are statistically significant. So in general, the areas where we're seeing grapes grown in Minnesota have low yan. That's actually a little bit too low, especially if you have a high Brix grape, like the Riparia-based hybrids. Iowa's a bit higher, and South Dakota is very high. If you live in South Dakota, I think it's safe to say do not add yan to your grape fermentations. Just for comparison, to answer the question we had earlier about white wines from the Finger Lakes, you can see that New York is quite low. And just because, you know, it's California, that's what we generally look to as our benchmark in the U.S. California is more about 200, which is right about average and safe and comfortable. So we see a lot more variety in this area. And again, These samples were provided to the researchers by folks at the respective breeding programs and universities. They are sourced from around the state, but I don't know where all of these samples came from, nor what the N is per state. So somebody said in the comments that South Dakota is probably a small sample size, and it probably is. But you have to think about the fact that in Iowa and South Dakota, we're often growing on old corn land, right? We're often growing on land that had row crops, and that tends to be the black dirt. So that might be a factor that we need to think about. Although we should see that in Minnesota too, in some cases at least. So anyway, interesting information. You can't make any rules from this, but it's something to keep in the back of your mind if you're growing in these states. So some more data. She broke it down by cultivar. If you look here, you can see her lacrescent data. She had 29 lacrescent samples from several states. If you look across the bottom, you can see from left to right, we're looking at Iowa, Indiana, and so these would be the Purdue research blocks, Minnesota, New York, and South Dakota. And you can see that lacrescent certainly varies by state, and as I mentioned, we know that we get variability by cultivar and variability by state, and also pretty much everything else. Climate, whether somebody sneezed in the vineyard, you never know. So Iowa is looking quite high here, Indiana a bit lower, Minnesota is low, New York a little bit higher, and so on. So I'm not sure if this number added to that 350 in South Dakota. That's looking pretty high. For comparison, for some of the work we're doing in New York, we actually have gotten quite a few samples of the crescent over the last 2 years, and we actually have been measuring ammonia, primary nitrogen, and YAN. So if you look left to right here, you can see that our growing sites were Geneva, Trumansburg, which is about 40 minutes south of Geneva down near Ithaca. It's a collaborator we work with. Clayton, which is our collaborator way up in the Thousand Islands near the Canadian border. Willsboro, which is another northern test site that's kind of in mid-state in between Geneva and the far north. And UVM, Vermont, worked with us in the Northern Grapes Project, and we've been getting grapes from them. So in any of these sites where I had samples from 2012 and 2013, you'll see those marked down on the bottom. So you compare by year, and I think this demonstrates pretty nicely that we certainly see year-to-year variance in some sites. You can see that in Geneva in 2012, all of my numbers were much lower on the lacrescent than they were in 2013, and those were two very different growing years, but not that different. So there's quite a bit of variability going on here, which is why it's hard to make estimates, and we'll talk about that in a minute. So you can see our range is there are quite a few that are below 150 actually in New York. Granted, this is a state where we normally see low numbers, but Vermont is a different state, and they are pretty low too. So only in a couple instances did we break that 200 mark. And a lot of this data is just observational, but since it's the first data that's out there for these cultivars, I wanted to just show it to you. Also, one thing to notice is that the ammonia is very low. We really would like to see between 25 and 50 milligrams per liter of ammonia in our total YAN, and we're not seeing that for any of these lacrescents. All right, so Amanda didn't have a lot of Frontenac Green data, so I'm not including the state-to-state that she had for lacrescent. But again, in our New York numbers, you can see again from left to right, sites in Geneva, Trumansburg, Clayton, and Willsboro, and 12 and 13 would be 2012 and 2013. And again, we see quite a variety. Frontenac Green is much higher than the lacrescent. We're up as high as almost 500 there in Geneva. That is huge. That's where I start to worry a little bit about having leftover stuff that other bad bugs can get into. And the low end is about 250. So in general, in New York, we're seeing Frontenac Green. That's pretty okay. I wouldn't add any YAN to any of these cultivars. You do see that in all cases except, well, we've got ammonia on the Geneva 2013. Our ammonia is over 50 parts per million, and most of them, it's in an okay range, but it's still relatively low considering the total YAN that we're seeing here. So we're seeing that in these two cultivars and cold-hardy cultivars in general, we tend to get more pan than ammonia. All right, so move on. A couple other whites I just wanted to throw in case you were interested. We have 2012 on the left, we have Prairie Star and Louise Swenson. And in 2013 on the right, we've got Brianna, Edelweiss, and two more examples of Louise Swenson and different sites. And the sites are then along the top there, Willsboro, Vermont, Geneva, and so on. And you can see that Prairie Star is okay. I wouldn't do additions for those. Louise Swinson, in all cases except for Willsboro, is all right, and Brianna and Edelweiss look okay too. So in general, we're seeing good total YANs, but again, we're primarily seeing low ammonia, so that's something to take into consideration. We move on to the reds. Here's some more data from Amanda Stewart's dissertation. You can see that, again, moving left to right along the bottom, Iowa, Illinois, Indiana, Minnesota, New York, and South Dakota. We're ranging from 500 parts per million down to, just under 300. So these all look pretty good across the Midwest and even in New York, where we tend to see low grapes. And I know for certain that these, I know that her samples are actually from a site down near Watkins Glen that's growing Frontenac. So this is just one site represented for New York, but in an area where we see chronic low YAN, this wasn't. Granted, it's only 1 year, but that's of interest. So if we could look at our comparative New York numbers, you can see that. And by the way, I should mention that Amanda's numbers are all from 2011. So it's a different year than the ones that I'm showing you from New York. The Frontenac numbers in the Northeast, looking from left to right, we have University of Vermont, our Willsboro trial site, Connecticut, that Watkins Glen site that she tested as well, and then our Clayton site up north. Mostly under 300, mostly not too low though, except for the Watkins Glen, which was, as you may remember, higher in the other one. A whopping 750 in our 2012 number from Connecticut. I'm not sure what they're doing there, but they certainly got the YAN up. That's a really high number. That's one to really be concerned about as far as keeping that wine stable and adding enough SO2 to keep the bad guys down later. And I see that Karine is noting that she's seeing similar numbers in Quebec, and so I'm glad to see that their research group is just seeing the same kind of stuff. All right, if we move on to Marquette, again, this is Amanda Stewart's data from across the Midwest, left to right across the bottom, Iowa, Illinois, Indiana, Minnesota, New York, and South Dakota. Again, a range from as low as under 200 for Minnesota to up to 600 for Iowa. And remember that this is going to vary by site as well as by state, but there was significant difference in these by state. So that's quite a range. I hope, if nothing else, you're starting to understand that you might need to measure your YAN because we're seeing such variability. And again, in New York, we just have a few samples from Vermont, Trumansburg, and Willsboro, so a more southerly Trumansburg site and the more northerly Willsboro site. Not huge. We're ranging from about just over 200 to about 300, so none of these need additions. So it's not as dramatic in the Marquette as it has been in the other cultivars, but we also don't have as many sites to test because there isn't as much of it out there. And just because we had a lot of the data, and I know a lot of people still grow at St. Croix in New York, we ranged from, again, along the bottom here, we have Geneva, Willsboro, Vermont, and Connecticut. Again, Connecticut seems to be a state that, at least on their side, has pretty high YAN, but you'll see that ammonia is still low in all of these. We're hitting most of that YAN with PAN, so we're not getting a lot of the simple nutrients there. All right, so a lot of people have asked how we measure, and I will give you some information about that now. The thing that I really need to stress is that there's no way to guess. You can guess what your sugar content is, you can guess what your acid is, and you've got, taste buds for this. There are ways to do that. Please tell me you're not guessing what your alcohol is that you report to the government, but beyond that, there are lots of things that we can kind of take a stab at if we've been making wine for a long time. You cannot taste YAN, you cannot smell YAN, and so you have no clue what's really in there. For years, we recommended that people use formal titration because it was fairly simple and something you could do in a winery. That's basically doing a titration with formaldehyde as we become more cautious about using formaldehyde and disposing of formaldehyde, especially in agricultural areas. This is not something we recommend any longer. You really need to have a hood, a fume hood, if you're going to do this the right way. And most wineries don't want to spend money on a fume hood, so and it's also not that accurate. It's really a guess. So endangering your health for a broad guess when you can send these things off for $35 doesn't seem like a good idea unless you really want to be pickled when they put you in the ground. So the other choices are spectrophotometric analysis, either with just a benchtop spec like the one you see here on the right. We've actually done courses here and taught people how to do this. You can get a good quality spectrometer for about $1,200, and it's very versatile. You can use it for things beside YAN, although YAN would be the thing that is most important. So if you are at that place and you would like to do your own measurements and feel fairly confident in your benchtop chemistry, that is something that you might want to consider. It's going to be a good investment. We do it a little bit fancier. We've got an automated system here. You can see my former graduate student, Mark Nisbet, who did a lot of the research I'm going to present in a minute, with his 96 well reader. This is what we use if you send samples to us during harvest. This is where your samples will go, and it's very accurate because it's automated and uses a very small sample. So for any of these, you need an enzymatic test kit. NOPA is the one for the pan, and thank you. And there is another one that we use for amino acids. So you actually have to run 2 tests to get your ammonia and your pan, but it's not too difficult. So when do you want to measure? This is a question we've asked. Can you use last year's number? A lot of people do, and we used to recommend that actually. Should you measure at crush? It's going to be your most accurate sample because you've got 100% of your grapes sampled if you crush and press and then test it. That's awesome. Some people find that this is hard to do. It's hard to line that up when you're doing 16 other things at crush and not sleeping and all that. How long can you wait for the results? If you don't have someone in your lab measuring it for you, if you send it out, you usually get return in 1 or 2 days. But do you want to keep your yeast sitting that long or your juice sitting that long? That's a question that a lot of people ask us. Can you measure before crush? Can you sample grapes in the vineyard? That's something we've checked out. You can do 100 berry samples. You probably want to do duplicate, juice them, and then use good sampling techniques. It's certainly an option and one that we can help you with. But our question really is, how early can you test your YAN? We know at Verizon at 5 to 8 weeks out or 8 weeks out, it's going to change a lot, but we decided in New York we wanted to test and see how early we could measure. So just to give you an idea, this is a conglomeration or a compilation of all of our Riesling YAN. We tested 60 sites a year from 2012 to 2010. You can see we have quite a range, but most of them are low. Most of them are below 100 milligrams per liter, which is quite low. So we're assuming that everyone in the Finger Lakes is adding YAN for the most part. I think there's one sample weight on the right-hand side that's above 200. Maybe 2, but most of them are low. So most people are adding some sort of nutrient, and we're afraid that most people are doing it prophylactically because this is hard to measure. So what we wanted to know is if we could measure it early, like 2 weeks early, and make some predictions. And just briefly, because I know a lot of you aren't growing Riesling, I'll show you the data. If you look in the upper left-hand corner, you can see our prediction on 2010 with our pre-harvest and our harvest. We've got about an R-square of 84.5, which is pretty good. That's a predictive R-squared, so we're actually testing the same sample from the same vineyard at the beginning and at the end. It kind of went a little haywire in 2011, which was a very wet year. We had a lot of rain between our 2-week measurement and our harvest measurement. You see our R-square went down to 53. In the lower left-hand corner of 2012, firmed up again around 66. So when we combined all this together and did a super huge model, you can see that we actually have predictive ability of about 70.3% there on the right, which isn't too bad. I mean, that made us confident enough that we could develop a model, I'll talk about in a minute, to help folks who want to measure early. They could just plug a number into the equation that we generated, or we can give you some prophylactic recommendations. If you're not going to measure, but you're growing Riesling in the Finger Lakes, we can give you a way to estimate. One thing that really surprised us, we had always, as extension folks said, well, measure the previous year and see if, that if you're not going to measure every year, at least measure from the previous year and base your decisions on that. But if you see up on the left-hand corner, that's 2011 that we tried to predict with 2010. And granted, 2010 and 2011 were very different years, as I mentioned. We had some rain on 2011 between testing and harvest. But our R-squared is a abysmal 11.6. That is not predictive. That is miserable. And then on the right there, 2012, predicted from 2011, again, our R-squared was 16.6. So despite what we have been telling folks for years as extension agents, predicting your yan by the previous year is not a good idea. So just because you measured it once doesn't mean you can assume that your vineyard's going to be at about that place forever. If you measure it 4 or 5 years in a row, you're going to be a little more accurate, but previous yan's not so great. So just to give you an idea of what can be done if you take lots and lots of samples, these are some of the, we actually developed several models. The one that worked the best for us was just this, the second one here. The MLR and the PLS were really huge, used lots of variables and weren't very useful, but that pre-harvest yan model at 69.5 prediction, that's pretty good, and that's what we're recommending for our Riesling growers in New York. And I want to stop at 50. Okay, so, excuse me, I'm getting time signals. So from this, we could calculate that if you're going to do a prophylactic addition in the Finger Lakes with Riesling, adding 96 milligrams per liter of nitrogen, so you're going to have to calculate the percent nitrogen of your additives, is going to be a lot more effective than what a lot of people do, which is just throw in 200 parts per million because that's the max, or the max recommended. So hopefully with calculations like this, we can get these down to a little more a reliable number. We also extended this, did the same thing with several other cultivars in our region, and came up with pretty good models as well. So while these are not useful if you live outside the Finger Lakes, and they are not useful if you're not growing these cultivars, it shows you this is possible, and explains why research is important in your region, because you can develop these with enough samples. You could develop these models in your region and have something to guide yourself with. One other thing I want to point out on this slide, if you look along the bottom where it says yan did not change significantly during the final 5 weeks before harvest, this really surprised us. We thought we'd see a lot of variability. The correlation between veraison and harvest was abysmal, and 6, 7 weeks out, but 5 weeks out, we started to see stability. And in the Finger Lakes, actually in all of New York, because these samples are across New York for these cultivars, things didn't change much for us over 5 weeks. And this, again, is over 3 years, including the year where we got a lot of rainfall in the last 2 weeks. So that's interesting. It's not what folks have seen in California, but we're looking at that. So what about hybrids? Well, it's a little bit different. In fact, we've had many polite discussions over glasses of wine with our colleagues at Purdue about this. They have found that in Frontenac Gris and in Ventura, which is a cultivar often grown in the Dakotas, that you actually see some increase in yan. The significance varies depending on the region, but you're going to see a small increase instead of, and this is from veraison to harvest. They found that yan didn't change a whole lot in the Crescent and Frontenac. There was a slight increase in both, but not necessarily one that was statistically significant. And then that yan decreases in Marquette, La Crosse, and Edelweiss. Now, none of these are things that we could say, well, it's always a 50% decrease from what you measure. But it means that we need to look a little more carefully at these cultivars and figure out at what point during the, 5 to 8 weeks of ripening that we're getting things become stable, and we would have to decide for each of these cultivars how early we could take a measurement. It's going to be different than the Riesling, but there is promise there as well. Just interesting to know. All right, one quick thing I wanted to mention before we finish up and get ready for some more questions. I told you in the beginning that the types of ammonia that we were looking at, the nitrogen sources, were either ammonia or amino acids. And there's been some interest recently in looking at individual amino acids. It's always been assumed that grapes in general were highest in arginine and proline because that's what we see in Vitis vinifera. But some work that, again, Amanda is doing, and some other folks are doing at Purdue, continuing her work with Christian Butzke's lab, and also a little bit of work that Ramon Mira Diorduña did here at Cornell, showed us the hybrid species are really variable, that we can't assume that arginine and proline are the 2 primary amino acids. In many of the Riparia-based hybrids, in Frontenac and Marquette, the Crescent and Frontenac Gris, we saw a lot of alanine, a lot of glycine, and then some arginine as well. But it wasn't just arginine and proline. In fact, we saw negligible amounts of proline in some of these. Why do you care? Well, for a couple of reasons. One is that we think, and we don't have great, a lot of research needs to be done on this, we don't have great information yet, but we think that some of the amino acids, the relative concentration of amino acids as they go through fermentation, give you different volatile compounds. And so part of what makes typical, aroma in a varietal character may actually come from the different amino acids that are in the grapes. In some of these amino acids that we're seeing in some of the hybrids, not so much in the Riparia base, but in some of the other interspecific hybrids, there's some amino acids like glutamate that actually have a gustatory flavor. They actually give you some savory tastes instead of just aromas. And if those are high enough, there may actually be a taste component there as well. And that's something that the Butzka group is looking at Purdue. So that's it's something to keep an eye on. The practical implications for these is that all of the recommendations for complex yeast nutrients assume that you're working with Vitis vinifera, and assume then that you've got adequate arginine and proline, and that may not be the case depending on which hybrids you're working with. So if you are interested in this and you know you need to use additives, you might want to have some detailed discussions with your suppliers about what's in their complex nutrients and what kind of grapes you're using them on. Lots of additives, in addition to YAN, exogenous tannins, lots of additives that we use have recommendations that are designed for Vitis vinifera. And so we know that you're probably going to need to do more bench trials and more experimentation to get the optimal use or the optimal fermentation parameters for your hybrids. So the take-home messages from all this, in addition to only use rehydration nutrients for rehydration, are that YAN is widely variable. We are dreaming if we hope that there will be an easy probe measurement anytime soon. I'm sorry about that. I wish there was. The only way to know what your YAN is to measure it, and you're probably going to have to measure, well, you are going to have to measure both ammonia and PAN. If you don't want to buy a spectrophotometer and you want to know what your YAN is, you're going to have to send it out, and you're going to have to send it out each year. And I realize that's an expense, but I would suggest that if you think it's important to measure your sugar, it probably is also important to measure your YAN, because a lot of the more subtle varietal character that you get from your grape is going to be predicated on what kind of nitrogen you have in there. So that's pretty important. Prophylactic additions in areas that have high, are using high YAN cultivars and have high YAN soils may result in significant excess, and that can be a problem because you might get microbial spoilage, and you might have ethyl carbamate, which at some point I suggest is probably going to be regulated in the U.S. And finally, we're interested now in looking at amino acid types and their concentrations, because they're quite different in hybrids than they are in vinifera, and they're quite variable across hybrids. So that's an area of new research that is pretty interesting, at least to me. I'm a flavor chemist, and so I like to know how we can fine-tune our flavors, and one of the ways may be by looking at individual amino acids. So I do need to thank Mark Nisbet. He did all of that Riesling work. Tim Martinson, our senior extension associate, helped plan and run that work. It was a long haul those 3 years. We got some really great data. Again, Dr. Amanda Stewart, who is now the enologist at Virginia Tech, was a student at Purdue when she did all of this great survey work. If you would like to get a hold of her thesis, I believe the electronic dissertation is published, and I would be glad to send you her email address, or you can look it up on the webpage at Virginia Tech. She will be glad to send you that because it is available. It's digitally published, so that's a nice big PDF. Cornell Federal Formula Funds paid for some of this work, so I need to acknowledge them as well as the Northern Grapes Project. And then finally, my email address is down there on the bottom if you have any questions that we haven't addressed. If you would like to get a nice overview of yeast nutrition and protection, Lallemont has this very nice free publication that I am sure they will send you, and or you could download from the web as a PDF. But it's a nice, user-friendly, overview of how this stuff works in layman's terms. Granted, it's a publication for their product, but they do pride themselves on doing good research, and I think this is a reliable place to look things up. So I think with that, that is the information, and I am going to turn it over to, Chris has got some questions for me. All right. So what does an average YAN test cost currently, Todd asked, and what kind of turnaround time can be expected? Both your cost and your turnaround time is going to depend on your lab. If you are in New York State, we do run it throughout the course of throughout harvest, and I believe our current cost is $35 if you're in state per test. And we try to do a 2-day turnaround. If you send it to us on Friday at 5 o'clock, you're not going to get it till Tuesday. But beyond that, we try to do a 2-day turnaround. ETS didn't query any of the other labs. Virginia Tech's lab, you have to call and ask for their specifics or look on the web. David Hill asked, has anyone had any success using the formal titration method for ballpark YAN analysis? Yes, you absolutely can use the formal titration for a very ballpark test, but as we mentioned, I would not recommend it unless you really like to breathe formaldehyde and want to pour it down your drain. If you don't have appropriate chemical laboratory facilities, including a fume hood and a way to dispose of formal waste, I would not recommend doing that in the winery. That is not something you want to play with, I don't think. Tim asked, how accurate do the pre-harvest samples have to be to guide ballpark additions? I'm not sure I understand that question. Tim, could you rephrase and send again, how accurate do the pre-harvest samples have to be? Do you mean sampling technique? Let us know. Send us another text and let me know what you mean. Did you say that a spectrophotometer for YAN could be purchased for $1,200? My husband said they were $6,000 to $10,000. Yes, LW, we did a price comparison about a year ago for a short course we were doing. We have the specs on 3 different YANs that are in that ballpark. If you want to send me an email, I will be glad to send you that information. And I'll also send you some information so that you can look and see what you need to, what specifications you want in a spectrophotometer for benchtop use. Matt asked, Anna Catherine, do you have any yeast recommendations for white wine fermentation that are less H2S troublesome than others? I found that QA23 has been very good, producing hardly any H2S, even with Finger Lakes juices. I would recommend that you look at, off the top of my head, no, because it's going to depend on what style you're going for. You want a different yeast for aromatic whites than you would for dry whites. All of that information is available in the yeast catalog. So Scott Labs, their catalog online has, I think they even have a fold-out chart where you can look for that kind of stuff. Inartis also has very good information. For Anchor, any of these companies, go through and read the specs, and that'll help. Okay, other questions? Keith says, do the same YAN rules apply for fruit wines, i.e. blueberry, apple, peach, and it doesn't depend on the yeast selected? Well, the yeast selected is always going to be a factor because some need higher or lower nutrients. So with your fruit wines, as with your grape wines, you're going to want to measure the YAN, and if it's low, you're going to need to decide what kind of yeast you're using and make your additions that are appropriate for both the yeast and your YAN level. Kevin asked, is there a method available for measuring YAN using spectrophotometer? Yes, if you email me, I can give you the information for that. What are YAN needs to second fermentation in sparkling wines? Oh, that's a good question. I don't know that answer, Kevin. We will hold that in reserve and see if we can find that out. I don't know. Good question. Michael Harney asked, have you seen too high of a YAN contribute to sulfide production? There is not, as far as I know, there is not academic scientific evidence to that effect, unless you get another sulfide-producing microbe in there that is happy with your extra YAN. So it's usually a low YAN problem. Tim says, do you have to get within 50 parts per million or 10 parts per million with your additions? If 30% variability is okay, then the measure doesn't, oh, okay, for your pre-harvest measurements. All right, so your analysis of YAN even is going to be, do you have to get within 50 parts per million? Tim, let's talk about this offline. There's not a simple answer to your question, so, but yeah, for your measurement, he's basically asking about measurements, how you're going to take your measurements to do a pre-harvest measurement and then extrapolate. I would recommend at least 2 100-berry samples and follow a good sampling mechanism, for instance, going zigzag through the rows and not using your boundary rows and that sort of thing. If you have questions about how to take a good 100-berry sample, email us and we will be glad to send that to you. But in general, yeah, you can't just take 3 berries and say this is enough. You need a representative sample in order to get the right data. So, all good. All right, thank you for listening, and send me an email if you have any other questions.
Okay, everyone. Thank you so much for hanging in there through our technical difficulties. We really appreciate that. I have put the schedule for the remaining webinars this season up on the screen. Please note that the March webinar is going to be on a Thursday and about a week and a half late from when it normally should be. Our speaker is going to be out of the country during the normal date, so we're pushing that back to March 20th. Anna Catherine, thanks for being a champ and hanging in there. We appreciate it. Thanks for sitting in my obnoxiously hot office, too, once the heat kicked on. And with that, I also encourage everyone to please go take the post-webinar survey. We really do value your input.